Yes. So I hope my screen is visible. If at any time you have any query or it is a connection failure from my end and I am not realizing it on my own, or my system is not prompting that for me, you are most welcome to unmute yourselves and intervene in that sense. Um, yeah, that is the same. It is going to be an interactive session. So if at all you have any doubts during the session, also maybe you can raise your hand. As soon as I finish my statement, I can attend to that, right? So let us keep it an interactive session so that it will benefit both of us, right? Because I have taught both kinds of sessions, the ones that are interactive and that aren't. And the latter is, I don't know, it feels less of less importance to me, right? So we'll keep it an interactive session. As you all might have already gone through the course description, and I saw that the people who had enrolled were from different, fields. So your specializations differed a lot. It should not be a problem because this is a beginner level course for neurobiology, right? If at all there are some biological terms for those of you who are from a non-biological background, I will try to, uh, even say out that concept in detail so that it is not a problem for you guys, right? So, yeah. Maybe I can just minimize this particular screen. Yeah. Apart from that, for a brief introduction about me, I am your course instructor, Arvind Kaur. I am currently a PhD scholar. I am in my fourth year, and I am here at IIT Kanpur at the Department of Biological Sciences and Bioengineering. And I am currently working in the field of neurobiology. So that field is related more to the neurobiology of disease and some mechanistic details about that. So this field was very new to me also when I enrolled as a PhD student, because my background is zoology. My bachelor's and master's is from zoology. And although we had multiple disciplines under that major also, I have never really studied neurobiology as a subject before, right? Prior to my PhD. So these basics I have also gathered within the last 4 years of my life. And it is pretty simple. It is just not a very uh, uh, actively taught subject, especially in bachelor's and all, unless and until you are majoring in neuroscience or cognitive science or neurobiology, right? So that is why I thought that if I have to begin with a module over here, before going into the research aspect of it, I would first like to cover the basics of neurobiology. So that is what this particular course is going to be about. And as again, you might have already gone through the flyer. Uh, the first lecture will be based will is the origin of neuroscience, right? So it is basically like a brief introduction or a brief history rather, or that is entailing to this neuroscience or neurobiology, right? So this particular session is more theoretical because it has a lot of history, it has a lot of names, because we are trying to understand to like I don't know if you have watched Big Bang Theory. It is one of my favorite sitcoms, right? So if somebody asks what is physics from Sheldon Cooper, he will begin in the beginning, right? So that is actually how a subject should be learned, not in not like that is not how it should be taught to somebody who is not that interested in a subject. But if you are willing to understand basics of neurobiology, you should begin at the beginning, right? So that some obvious questions that might come up in your head, or that should have come up in our minds, will be discussed over here, right? That okay, back in 1700s, people with such limited resources could even think in this direction, right? Now with technology at our hands, still I am not probably able to think in that direction. So that is also to instill your thought process, right? So that is why history, understanding the history of anything, to understand the ethology or etiology of a word, it is important, right? Yeah. So, having said that, we can begin with our course. I would also like to mention over here as to where I will be gathering my teaching material from. So, it is from two primary books. For today's lecture, I have gathered the material mainly from Principles of Neuroscience by Candle et al. It is very, it is like a neurology Bible, right? No, the clinical neurology, the basic neurobiology Bible. So it is a very sort of a book. It is easy to understand, and this lecture has been prepared majorly by chapters from this book itself. Apart from that, there's another book that I'll be referring through my course, which is The Student's Guide to Cognitive Neuroscience by Jamie Ward. So he is also a professor at Cambridge, if I'm not wrong. And that is another book that is easy to follow, and it has great clarity regarding concepts of neurobiology, right? So, apart from this, wherever any other resources other than these two books have been used, I have mentioned them in the slide itself, right? So you can find that resource and you can go and you can read through from that also if you are interested, right? Yeah, so that was basically the introduction. Now let us jump on to the actual lecture, right? I'm still really disappointed regarding the link, how there was such a infusion, right? So that most of you did not get the link, I think, right? Anyway, first, whenever we are beginning anything, we should always understand the etiology of the title itself, right? Etiology as in the origin. What is the meaning of the title? What is the origin of that title, right? So over here, we will study two words that are often used interchangeably, but we might also sometimes use them interchangeably, but once we should understand what is the major difference between these two, right? So these two are neuroscience and neurobiology. So when we are talking about neuroscience, right, what is neuroscience? It is the study of the nervous system, right? So that is also what neurobiology is, right? So what is so different in neuroscience, right? So it is a multidisciplinary field that integrates numerous perspectives from biology, psychology, and medicine, right? Again, even up till now, it feels like it could be very similar to that of what is neurobiology, right? So, because neurobiology also contains, the medical and the biological perspective, and to some extent, even the cognitive and psychological perspective, right? But the difference comes over here that this neuroscience combines several sciences such as physiology, anatomy, molecular biology, developmental biology, cytology, psychology, physics, computer science, chemistry, medicine, statistics, mathematical modeling, to understand the fundamental and emergent properties of the neurons, glia, and neural circuits, right? So when you talk about uh, neuroscience, for instance, you would have, even if you are not from a biology background, and especially if you are from an engineering background, you might have come across the terms neural networks, right? So are neural networks networks of neurons? In literal sense, yes, but in algorithm sense or in terms of engineering, that is not what it means, right? Those are mathematical models, right? So, like, neuroscience entails a much wider, it is a much wider umbrella, and it basically covers a lot of disciplines under it, right? So that is this point is a major difference between neuroscience and neurobiology. Whereas, as we were talking, what is neurobiology? It is a sub-discipline of biology that focuses on the cellular and molecular aspects of the nervous system. Now, this, apart from the cellular and molecular aspect, obviously, it can also be related to the anatomical, physical, physiological, morphological, aspect of the nervous system also, right? So that is always a given. Also, it investigates how the neural cells function and interact to form the complex networks that underlie the brain activity, right? So that is what is the focus of neurobiology, right? Neuroscience, however, is also focused on understanding how the mind is working, right? How the brain is functioning is more of a neurobiology perspective. How the mind functions, or how we think, how we calculate, how we express emotions, etc., are more of a neuroscience topics, right? So now I'm talking about brain and mind as separate, entities, right? So we will soon come to that also, and we will discuss how these are different, how these are, similar, and why are we basically calling them as two different units, or why should we call them as one unit, right? We will come and we will discuss that also. So, neurobiology is basically the study of the brain and the nervous system, which are the cells and tissue that generate sensation, perception, movement, learning, and emotion, and many of the functions that make us human, right? So, that study is basically under neurobiology, right? Now we come to the brain and the mind, right? So, as I was saying, there would be some difference between the two. That is why we are referring to them as two different entities, or two different, objects, right? Okay, so the mind is actually the realm of thought and consciousness, while the brain is the thinking machine, right? So, what does this mean? Okay, just a moment. Yeah. Okay, one of your fellow batchmates tried to join. I am not sure whether she could. Okay, anyway. So, what did we say? Mind is the realm of thought and consciousness, right? So, it is at a much bigger level, right? It is where thought is occurring, it is where we are conscious, our subconscious is lying, right? But brain is the thinking machine, right? So, for instance, we could take another example that is of this computer, right? So, in this computer that I am using to basically, converse with you and to present this particular PowerPoint presentation, right? I would have to say that the hardware of the computer, right? The keypad, the monitor, the mouse that I'm using is all like the brain, right? So these are physical entities that are doing work, right? But the actual softwares inside the Microsoft 365 package that I have, that is actually doing stuff, right? Or that is actually giving me a platform to do stuff, right? Or the Chrome, the Edge, the different the different explorers that I have, right? All this could be, homologous to the analogous to the mind, right? So that is like one example that maybe you can relate to, right? So brain is the tangible part. Now, what does tangible means? It means that something that you can touch. So brain is, tangible, that you can feel the brain, right? It is like the hardware, as I said. it is a bustling metropolis of neurons, right? So there it is filled with neurons, right? So there are so many neural cells that are present within the brains, right? Within the brain, with more connections than there are stars in the Milky Way, right? So now this is such a thoughtful statement, such an intriguing statement in some terms, right? So there are more connections in every one of our neurons than there are stars in the entire galaxy, right? So that is actually a very intriguing statement. Okay. So when we talk about the brain, I'm sorry if I'm not realizing, but I do have sinusitis and it is bad today. So if at all you cannot understand what I'm saying, please let me know. Uh, I don't know what I'll do, but maybe I'll try to keep it in check. Right? Okay. So brain is actually the tangible part, right? We have already read this in the previous page also. And this is another fact that we have already studied. I think it was a duplication of this point that was done over here. Yeah, yeah. Okay. So from this, okay, so it is a similar example, right? So imagine that your brain was a super advanced computer, right? Instead of silicon chips in your computer, right? So for instance, in your phone, you have those Snapdragon chips or the Exynos chips or the iOS, right? All these various chip systems you have, right? Instead of that, consider your phone to be your brain and the chip set to be the mind, right? So instead of silicon chips, it is built up of, oh, but chip is also tangible actually, right? So chip is a part of your brain, right? Not the mind. So it is made up of billions of tiny cells called neurons, right? So that is one analogy that they have given. Now, if your brain is actually the hardware, you might think of your mind as a software, right? But the mind is a slippery concept, defying easy definition or measurement, right? So, how to explain to somebody what is a mind, right? So, if I have to explain to you what is mind, I will also have to explain to you what is brain first, right? Or I will have to talk about both of them simultaneously. Otherwise, I will not be able to get my point across. Right? So it is because it is not tangible, it is not an existing entity, it is a concept. Right? That is why it is a little difficult to explain that concept or to measure that concept also. Right? So when we talk about, hold a moment. Yeah. So when we talk about the mind, we are diving into the realm of consciousness, thoughts, and emotions, right? So it basically deals with the thought process, the thinking process, the memories, right? The emotions, etc. Right? It is a part of us that experiences the world, that feels joy, pain, that ponders the meaning of life, right? So all done by the mind, the consciousness within our brain, right? So our mind is where our inner monologue lives, right? Where we dream up wild fantasies and solve complex problems, right? So although brain has multiple compartments, right? Even till date, we don't know where exactly memory is stored, right? So we have listed a region. It is known as a hippocampus, right? That is where memories are formed. Sometimes that is also where those neurons are also where memories are stored, but actually it is not like a tangible thing that is going and getting stored, right? It is a connection of neurons, that connection is being remembered by the brain, right? So it is a very complex state. Okay. And we will try to decomplexify this entire thing throughout this course, right? I hope so far there was no problem per se, right? Okay. Yeah. So the mind, however, is intangible, right? So it cannot be touched, it cannot be felt, it doesn't exist in, in it is not particularly in existence, right? It is there, but uh, you can't touch it, you can't access it. So, when we talk about brain versus the mind, right, we are basically talking about the physical versus non-physical, right? So, brain is tangible, so you can hold it in your hand, right? Obviously, we are not going to hold it in our hand, but yeah, we know that the brain is there, right? We have seen probably brain in a jar. I work with mice, so I have seen the brain of mice at least, right? So, you might have seen it in movies, you might have seen it in pictures, etc. Right? So that uh, that part is there, right? The mind, on the other hand, is more abstract, right? So abstract anyway is something that is a little beyond explanation, right? You can't put your thoughts under a microscope or weigh your emotions on a scale, right? So that is true, right? You cannot quantify your emotions, right? I am feeling like I can say that my brain is maybe uh or I can say the mouse brain is around 50 millimeter, right? Or around 5 centimeter. But I cannot say the mouse is feeling, say, 12 grams of pain or 12 kgs of pain or 12 liters of pain, right? So that is something that is not, it is abstract, right? It is arbitrary. It is not measurable, quantifiable, right? Why? Because it is non-physical, right? Also, when we talk about subjectivity and objectivity, right? It differs between whether we are talking about the brain or we are talking about the mind, right? So the brain activity can be observed and measured using scientific instruments, right? So you might have heard of people who are going from for EEG, for instance, right? Electroencephalography, right? So they get a brain wave pattern. From that, the doctors and neurologists, they try to decipher what is up with your brain. Or some people will go for an MRI to see like what is the gross morphology of the brain, right? CT scan to look at the morphology again. fMRI, wherein they will basically look at which area of the brain is active or which area of the brain is getting an epileptic shock, something like that, right? So there are multiple instruments that are available that can be used to observe the brain's activity, right? Multiple instruments are there, and we will probably discuss a few of them in one of the lectures, right? But when we talk about the contents of our mind, right, our thoughts, feelings, perceptions, they are inherently subjective, right? Only you have direct access to your inner mental world, right? So even if I go to a psychologist or I go to a psychiatrist, right, can they tell me yeah, so if they check my neurotransmitter levels, again, that is something that is tangible. They can see that, okay, I think that the dopamine or the serotonin is lacking, right? It should be in this range, it is in this range. That is still quantifiable, right? But particular feeling, right? How sad is she feeling if there is drop in serotonin or drop in dopamine? That is not quantifiable, right? So, only a person has access to his or her own exact status of the mind, right? That mind cannot be quantified. It can be perceived by others maybe a little, uh, but For instance, okay, let me give you another example, and I don't know how many of you know this or not, right? So the color that everybody observes is very subjective, right? So, for instance, I have a blank black and white slide. So, over here, I cannot show you any color. But for instance, I had this pouch, right? So, I hope you can see this pouch. So, it has multiple colors. It has a pattern and it has multiple colors, right? So it has a red kind of outing or piping or an outline, right? So over here, what I want to tell you is, according to me, this is red, right? This part is yellow and this part is green, right? And most of us have will have that same thought, right? But some people, I'm not going into whether it is teal or it is sea green or it is blue or it is green, right? I am saying that it is some version of green, right? This part is some version of green. But to some minds, this part, because it is a little ambiguous between green and blue, might appear blue, right, instead of green, right? Also, the color red that I am seeing is very subjective to me, right? It might be a little different than your experience of seeing this color, right? So, it is not always the case that the perception of color is same for everybody, right? So, this red that I am observing might be a little different than the red that you are observing. Not because you are observing it through a camera over here, even if you were sitting right over here beside me, the perception of color would be very, very subjective to individuals, right? So you will have one kind of image about a red, and I will have another kind of an image about a red, right? So that is also a little different, right? So uh, that is just another pointer to tell you that this subjectivity and objectivity basically exists, right? And the mind is majorly subjective. Okay. Also, we can map brain structures, measure electrical activity, and even watch neurons fire in real time, right? Some of the techniques that I mentioned, fMRI, PET, etc., you can do these things, right? Or patch clamp, etc., you can measure electrical activity. EEG also measures electrical activity in a sense, right? So that is possible. But measuring the mind is a whole different ball game, right? How do you quantify a feeling of deja vu or the intensity of a childhood memory, right? I can say that the dream that I had was very bad, right? But is my level of very bad the same as your level of very bad? Maybe it was very mild for you. Maybe it was extremely painful for you if you would have the same dream, right? So that is different. That is what we are trying to discuss over here. Also, when we talk about the functionality and purpose of each of these entities, the brain and the mind, right? What does that mean, right? So, the brain's primary job is basically to keep you alive and functioning, right? So, what is the primary job? It will regulate your heartbeat, it will control your movements, it will process sensory information, right? If I touch something, or I will basically remove my hand from there. If I am seeing a snake coming towards me, I will probably run away, right? Or I will put something on it so that it cannot attack me. Uh, and it is controlling the beating of my heart, the movement of my diaphragm, due to which I can breathe properly, right? So all this is basically the job of the brain, right? To keep you moving, to keep you functioning, right? It is like a battery in the computer, right? So it is very, very important for you to function, right? So the mind, however, deals with higher level functions, right? Like reasoning, creativity, self-awareness. So, for instance, uh, say I am very good at math solving, complex math problems. And there is another person next to me who is poor at solving math problems, but that person is highly creative, right? So the kind of that person is an interior designer, and the kind of combinations that the person can put together is way beyond my league, right? So all these things, right? That is not because he has probably a better developed, art or creative area in the brain, and I have a less developed creative area. I have a more developed area that will do mathematical functions. So that is not true, right? Some exceptional cases can be there, but generally that is not true, right? So it is how your mind is, how it depends a lot upon your experiences, right? So how you are shaping your mind, the brain is very plastic, right? So there is neuroplasticity that exists, right? So the way you shape your mind, or the kind of experiences that you have, will shape your thinking, your reasoning, your awareness, your creativity, etc. Okay. So the brain, like any physical organ, ages over time, right? Cells will die, connections will weaken, cognitive decline will also set in eventually, right? So we will eventually, for instance, I'm good at driving, my motor functions are fine right now, but maybe when I'm 90, I might not have good motor functions, right? My reflexes might not be that good. So I might not be good at driving at that time, right? So obviously neurons are not dividing, so some of my neurons will also be dead. Because of that, connections will alter, they will weaken, right? Neurotransmitters will be lesser in my brain at that time, right? All these things will happen. But in case of mind, right, it can continue to develop and grow throughout life, right? For instance, I said the neurons don't divide, right? Generally, the neurons don't divide. There are some exceptions that are there. There are one or two hotspots that have been recently discovered wherein neurons can also divide, right? But it is uh, not throughout the brain, it is at one or two specific locations in the brain, right? So mind, however, can always continue to grow and develop throughout life, right? So a neuron once formed will remain like that, right? It will just maybe alter its connections, how it is interacting with this neuron right now. Next, it will interact with the other neurons. So it will, that will change connections and all. But in case of mind, it doesn't require any, substrate other than, experiences, knowledge, etc. So it can always continue to grow throughout life. You can learn new skills, you can gain wisdom, right? So the older you get, the more wisdom or the more experiences you get, right? So for instance, 10-year-old me would not have been aware of probably, you know, a hospitalization experience, or an experience regarding a close friend drifting off, right? These are experiences that you gain with age, right? So, you can also learn new skills. Again, 10-year-old me did not know how to drive a car. Now I know how to drive a car, right? I can expand my consciousness well into old age, right? Still I'm learning. Right? I am doing my PhD every day, or every week at least. I'm finding something new related to what I'm doing, right? So it is always, the knowledge is keeps on adding, right? So that is my mind that is growing from that knowledge. Then, in this relationship, while the brain and mind are distinct in many ways, right? We talk many ways in which they are so different. They are also intimately connected, right? It is not like brain can function as a separate entity and mind can function as a separate entity, right? It is not the case. They are well interlinked with one another, right? So is their function. Okay. Yeah. So, from a neuroscientific perspective, the brain and mind relationship is all about correlation, right? So, when we think certain thoughts of or feel certain emotions, specific areas of our brain will light up, right? So there have been multiple studies, even on humans, and non-invasive experiments, right? So there's nothing invasive. You don't really now, at least you don't really have to do surgery to, have these kinds of observations, right? There are simple tests that you can do. You can inject a tracer and you can follow it. You can see that which area of the brain it is going to when you are playing the music, which area of the brain it is going to when you are making the person solve a math problem, etc. Right? So that is possible now. Your thoughts and emotions can actually change your brain structure and function, right? So over here, this is another way that they are so interconnected. So we are thinking something. Because of that, my neurons are firing in a certain way, right? So because of one, it is like I open a software in my laptop, due to which, say, my laptop starts, heating up. So it is a physical response to a software, right? So similarly, uh, I am thinking about something due to which my neurons are firing in a certain way, due to which probably, I will form a new memory or I will form a new connection between neurons, right? Which will alter my brain structure in a certain way, right? So, sorry. Oh, I'm so sorry. Right. It is getting a little harder for me to speak now. So we will see how long we can go. Otherwise, because I have not taken any medication as of now, it is very new, the cold, right? Otherwise, I will continue with this particular part on Sunday, right? So instead of 12 to 2, probably, if I am unable to finish this part today, I can take that session a little longer, maybe till 2:30 or 3, so that we can get done with the session, right? Also, probably by then, all that link thing will also be sorted, right? So, all of you could join by that time, right? So, yeah, okay. So, yeah. So when you learn a new skill or you overcome a fear, you are literally rewiring your brain, right? So I'm sure that this is a concept that you are aware of by now, right? That there is some kind of chemical changes, structural changes that go on in your brain when you are thinking, when you are learning something, right? Because that is going to create a muscle memory, right? You might have heard this term muscle memory, right? So muscle is a physical entity, right? So creating a muscle memory means that you do something such so many number of times or so repeatedly or repetitively that it will, by default also you can do that, right? You don't really have to access the software in that case. You don't really have to go to the mind, or it is so quick that the mind responds like that, right? In a fraction of a second, in a fraction of a millisecond sometimes, right? So because it has come into your muscle memory, right? For instance, if I want to tie shoelaces, I will not have to pay attention to it while tying, right? Maybe even while talking to somebody or something, I can still tie my shoelaces, right? Similar is driving, right? Earlier, when I was learning to drive, I had to think about a lot of things consciously, right? After you start driving and you get used to driving, particularly, you have to be aware about your surroundings, but you are not constantly thinking about, okay, I have to leave the paddle this much, press the paddle this much, clutch should be just pressed this much, I need to change this gear, this should happen. So all that is not consciously probably going on in your head, right? You are just doing it because it is a muscle memory, right? So that is what happens. Then in this course altogether, we will basically follow or focus on a unified theory, right? So, as the name suggests, it means that we will talk about the brain and mind in collaboration with one another, right? So, unification of the study of behavior, which is the science of the mind, and neural science, which is the science of the brain, right, is what is called unified theory. So, such a unified approach, in which mind and body are not viewed as separate entities, rests on the view that all behavior is the result of a brain function, right? So, that is what unified theory is. What we commonly call the mind is a set of operations carried out by the brain, right? So the brain processes underlie not only simple motor behavior such as walking, eating, but also complex cognitive acts and behavior that we regard as quintessentially human, like thinking, speaking, creating works of art, right? So that is very specific to humans, right? That is what this line means, right? Even there, only the mind is not involved, the brain processes are also involved over there, right? So this introduction basically brings us to a few questions, right? What are these questions in general uh, that we have as a group together, right? As a group of humans, these are some questions that are some of which are open questions, right, in the field of neuroscience or neurobiology, right? So, first is a particular mental process carried out in specific regions of the brain, or does it involve brain as a whole, right? So there are actually very interesting theories regarding this also that had come up in the early 18th, 19th century, and we are going to discuss about the people who brought forward those theories and what they were, and where does the theory stand right now, right? So what is our understanding right now, right? So this is the first question. The second is, if a mental process can be localized to discrete brain regions, what is the relationship between the functions of those regions in perception, movement, or thought, and the anatomy and physiology of those regions, right? So, this basically talks about the relationship, exact relationship between the brain and the mind, right? So, if I am thinking something, which brain area is lighting up, right? That is one thing. And we have often uh, you know, uh, looked at that also. Maybe if I am trying to recollect something, my hippocampus might light up, right? There might be another area in the cortex that might light up, right? Because I am remembering some particular kind of a visual or some particular kind of smell. So my olfactory cortex will light up, right? So, how these areas that might not be adjacent to one another, how are they communicating? How are they altering physiologically when we are accessing these memories, etc. Right? So, all this relation between how the mental processes are going on and how brain is responding to that is another question, right? That we are trying to explore. Also, are these relationships more likely to be understood by examining each region as a whole or by studying individual nerve cells? Right? So, now this again is a very abstract kind of question, right? It depends on what feature or what character you want to study, right? If you want to study, for instance, uh, how particular neurons are, how interaction of neurons is occurring, or how does neurotransmitter help in transmission of signal, or how is action potential generated, right? Over there, individual cells might be a good option or a better option to study, right? or multiple individual cells brought together so that they form a connection might be a better option to study, right? But if we are trying to understand that if we are giving this particular stress to this particular model or this particular mouse, is the mouse in a depressed state? Is the mouse, still active? Does the mouse have an epileptic phenotype? So in that you will have to study the whole system, right? Studying individual cells will not do you any good, right? So it will depend upon what aspect you want to study and what is the character that you want to study, characteristic feature, right? Okay. So, okay. So, from this point forward, we actually begin with the history of neuroscience and neurobiology, right? So, one of the oldest actually mentions about brain has been by Galen, right? So Galen was a Greek physician who existed in the second century, right? So he was a Greek physician from second century who basically proposed that the nerves convey fluid that is secreted by the brain and spinal cord to the body's periphery, right? So at that time, what he said was, for nerves, like he is talking about nerves similar to blood vessels, right? So blood vessel is taking fluid from the heart to the other body parts, right? And from the body parts back to the heart, right? So similarly, he thought, because if you try to visualize blood vessel network looks very similar to the nerves network, right? So that is what he said at that time, with whatever observation and knowledge he had, that nerves convey fluid secreted by brain and spinal cord to body's periphery. So this means that he, even in second century, believed that brain is the seat of the mind, right? That brain is controlling the body's peripheral organs, right? And spinal cord is also controlling the body's peripheral organs, right? So he was aware about the concept of the nervous system, right? That this is a system that is controlling the entire body, and how it is controlling through nerves. And what do these nerves do, or what are they? They are basically networks that are carrying fluid that is secreted by these two organs to the body's other parts, right? Then his views dominated the Western medicine until the microscope structure revealed the true structure of the cells in the nervous tissue. Right. So once the microscopes were actually developed, one could look at cells, and once one looks at cells, one can see that these are not hollow conducting tubes like vessels, right? So vessels are hollow, they have a lumen within them. It's a neuron is not like that, right? So in the upcoming lectures, we will study what a neuron looks like also, right? So that is not what a neuron looks like, right? It is nowhere close to a blood vessel. So it doesn't have that hollow lumen. How will it basically take how nerves, how will they take fluid, right? If they don't have that hollow lumen within them, right? So also they have multiple different kinds of features, right? They have some kind of sheath on them. They have some kind of axonal feet that are there, dendritic feet are there, etc. Right? And there are also uh, the nerves are not continuous, right? So there are distance between the cells, right? So if it is not continuous, then will the fluid not leak out, right? There might be another way through which they actually communicate. All these questions arose later when these basically nerves were observed under a microscope, right? But until then, a lot of people actually believed what Galen said, right? So he was influential, he was a physician at that time, and a lot of people actually believed what he believed in, right? So the nervous tissue did not become the subject of special science until late 1800s, right? And these two people, the Italian scientist Camillo Golgi and the Spanish scientist Santiago Ramon y Cajal, produced detailed, accurate descriptions of the nerve cell, right? So in the late 1800s, these were the people who are basically, our understanding of neurons basically started because of techniques developed by Camillo Golgi and the fabulous utilization of that technique and fabulous observation skills of Santiago Ramon y Cajal, right? They both actually got the Nobel Prize also, right? So we will read in detail also what each one of them had done, right? Yeah. So in case of Golgi, this is Professor Camillo Golgi. He developed a method of staining neurons with silver salts that revealed their entire cell structure under the microscope. Right. So why does one need to stain neurons? Right. So it is an open question. I don't know how much you guys will be a part of the discussion, but I will keep trying. So it is an open question. Why do you think Golgi had to develop, or why was it so relevant that he developed a method of staining the neurons? Anybody? You can give it a try, right? There's no wrong answer. Okay. Anyway, I will still discuss it, right? So, why do you stain any tissue, right? Because you want to differentiate the structure within the tissue, right? So every time you stain something, you have something in mind because of which you are staining this thing, right? So for non-biologists, maybe I'll just think of something, right? By the time I make people who have a biology background understand it a little, right? So, the people who have a biology background, right, you might be aware of simple staining techniques such as, say, HNE, hematoxylin and eosin staining, right? In that, there will be an acid dye and a basic dye, right? And they will differentiate the cytoplasm and the nucleus, right? So, one will stain the cytoplasm, the other will stain the nucleus, right? So, how is it relevant? Because when we are looking at a cell like this, we will not be able to make out we will have to have really good lenses in our microscope to actually look at the details of the cells, right? But if we stain them, right, it is easier to look at, right? They are not transparent anymore. The nucleus is distinct now, right? It is of a different color now, so they are more easy to look at, right? So you have purple nuclei, you have pinkish colored cytoplasm and other tissue, right? So that is why staining is done to differentiate tissues, right? For people who are from a non-biology background, you can consider a case that, for instance, a person met with an accident, right? And the person died, right? Now, this person's uh, uh, person had, say, a neurodegenerative condition, right? And the doctors that were treating this person wanted to do an autopsy on the brain, right? To understand like which particular place that they knew what disease this person had. But till now they don't know exactly what all regions this disease can affect, right? So there were some, EEG recordings, some CT scans that were done, but now, because the person had died, And before dying, he had agreed to this autopsy, or his family members did agree to the autopsy, right? So that the science could advance, so that that disease that this person suffered from doesn't, affect, or at least is it fatal for most other patients for of that disease, right? The person might have agreed, due to which an autopsy on the brain was done. So now we have an individual who is dead, whose brain is being autopsied, right? So a person could take out the entire brain and try to look at it, right? Do you think they will be able to, look at the neurons, the neural cells? Do you think that is possible? So the answer is no, right? So that is not possible, right? Now, oh, how can one look at the neurons, right? I wanted to see that whether the neurons had the myelin sheath or not, right? It's a part of the neuron. Whether their dendrites were normal, like we see in cases of in normal brains. Is it like that, or is that where the difference is occurring? How will one see that, right? So one aspect could be, or one thing that you could have thought of is that we can cut the brain into smaller slices or thinner slices so that we can look at it under a microscope. Right? Brain we cannot put under a microscope. It is a big entity. Microscope lenses are for much tinier objects, right? So if we slice the brain into very thin, pieces, we can maybe look at it under the microscope, right? So that is possible, uh, and that is doable also. But when you look at brain under a microscope, will you be able to make out where the myelin sheath is? Like it is not the kind of structure that even you can see you can distinguish in a thin slice in a microscope, right? Or the cell body of a neuron is something you cannot see. You can see the gross morphology of the brain slice, right? You cannot look at the individual cells, right? Because there are so many cells. Can you spot neurons in that? It will be very difficult, right? Why? Because there are several neural cells that are there. There are glial cells like microglia, astrocytes. Together they are more abundant than the neurons. So maybe the part that you are seeing is a glial cell part. Maybe it is not even a neuron. How will you know? Right? So for that, staining is done. Right. So what is the purpose of a stain? It is basically going to tag a particular kind of cell, right? So it will, the stains are developed based on some biochemical acids, right? So biochemical reaction will go on. If I, for instance, if I stain a slide with a stain that is known as nuclear fast red. As the name suggests, it will stain the nucleus, right, into a reddish pinkish color, right? So if I drop that stain on the slide that has a section, right, it will stain all the nuclei, right? Why? Because some kind of a reaction went on between the stain that I have dropped and the nucleus, so that then only the nucleus retained the color, right? So it is based upon principles of chemistry, right? And the principle for each staining varies, right? It is different. But the crux is that it will basically staining is done to identify individual parts of a cell or individual parts of a tissue, right? So over here, there was for the first time, people could actually distinguish and look at only neurons because of a stain that was developed by Golgi, right? It is known as Golgi-Cox stain. It is still actively used, right? Also, it is known as silver staining or silver nitrate staining. So one of the silver salts that he used was silver nitrate, right? So it is a very, very effective stain, right? It stains only the neurons, right? Not the other cells. I hope that, yeah, I thought there was a disconnection of the meeting. I think that is not the case, right? So that is what Golgi did, right? For the first time, he developed something through which we could only look at the nerve cells or the neurons, right? So this is what it looks like, right? So after the silver nitrate staining is done, this is what we see. So these are basically neurons, right? If we just look at the brain, we will not see these in a different color, right? We will see everything in this color only, this background color. Right? So it is very difficult to make out that, okay, this is maybe the cell body is bigger, so it is we are able to make that out if we are used to looking at such slides. But these connections definitely we can't look at, right? So he could see clearly that each neuron typically has a cell body and two types of processes, right? They are either the branching dendrites at one end and a long cable-like axon at the other. So these are the two processes they are talking about. This is a long cable-like axon and these are the branching dendrites, right? Similarly over here, this is the axon, this is the dendrites. So this he constantly saw across all neurons, right? Then, based on this, he came up with the reticular theory. Okay, actually, the reticular theory was not his theory. He was the one who popularized this theory or supported this theory. So, what is the reticular theory? It states that the nervous system is composed of a single continuous network of cells rather than separate nerve cells, right? So, if you look over here, it says that this is a single network of cells, right? It is not that this is a separate neuron, this is a separate neuron, these are dendrites from this particular neuron, these are dendrites from this particular neuron. No. Everything is interconnected, it is what he said, right? Or what he supported. So this theory was actually proposed by Joseph von Gerlach in 1871 and it was later popularized by Camillo Golgi, right? This reticular theory. Now we know actually that this theory is incorrect, right? There is another theory that has been proven after this, which is the neuron doctrine, which we are going to very study very soon, right? So that was Golgi, right? Now, this is another fellow who, along with Golgi, only received the Nobel Prize for advancements in neuroscience, right? Now, this person was, he was one of the best scientists that has existed because just by a simple lens, not even the fancy microscopes that we have right now, wherein and the fancy staining techniques that we have, wherein we can stain, different parts of the neuron with different colors, etc. Right? Just with that simple silver nitrate staining and with a simple lens microscope, not even a compound microscope, right? Ramoni Kahal was able to, form a doctrine that is true today. We are going to study that very soon, the neuron doctrine. And he has made fabulous observations, so many of which are true till now, right? Plus, this is an actual drawing that at that time there were no cameras attached to these microscopes, right? So, what he used to do was he used to put a slide, he used to look at the slide and then he used to draw on paper what he's seeing on the slide. And there are beautiful drawings of the brain sections by Ramoni Kahal. There are hundreds of them that exist, and like you will really appreciate it once you look at it. Okay, sorry. So in the course of, I have not read the first part also. Using Golgi's technique, Ramoni Kahal discovered the nervous tissue is not a syncytium. Syncytium meaning it is not a continuous web of filaments, but a network of discrete cells, right? So using what technique was created by Golgi, this is what he discovered. In the course of this work, Ramonika Hall developed some key concepts and much of the early evidence for the neuron doctrine which says that the principle that individual neurons are elementary building blocks and signaling elements of the nervous system. This is what we know about it today. What is the basic functioning unit of a nervous system? It is a neuron, right? That is the neuron doctrine that was first given by Ramoni Kahal, right? Based on his observations and drawings, right? So he was not in support of the syncytium or the reticular theory that existed, and which Golgi also supported, right? He gave another theory which came to be known as the neuron doctrine, and this is that, right? Also, just so that you can appreciate, these are basically copies of uh, actual drawings of the brain that was done by Ramoni Kahal, right? These are some actual drawings. Okay, I don't know like how relevant it is, but again, referring to Big Bang Theory, if you guys are as big fans of that particular sitcom as I am, you will remember that Sheldon had asked his assistant to fetch for a gift for his girlfriend Amy, who was a neuroscientist in that show, right? And one of the things that the assistant of his had brought was an original painting of Ramone Kahal, right? An original drawing by Ramoni Kahal. It looked something like this, right? So this is that fellow, right? And there is a compilation of his drawings that is in this book, which is known as The Beautiful Brain, right? Which has the drawings of Santiago Ramon y Cajal. And this is a fabulous, fabulous book to look at. If you get a chance at any bookstore, or, any friend of your, professor of your has this book, I would really recommend that you go and flip the pages. You will be in awe, right? Because these are all hand drawings. And yeah, it is fabulous. So, yeah, that brings us to another scientist who is Ross Harrison, right? Uh, Before we move on to how other people have also supported this neuron doctrine, and they have actually provided tangible proof for this neuron doctrine, due to which this is the widely accepted theory now, right? They have disproven the reticular theory altogether. Uh, I would like to point out that despite Santiago Ramon Cajal and Camilo Golgi having different views, opposing views actually, regarding how the organization of the brain was, right? One was in favor of the reticular theory, the other was in favor of the neuron doctrine, right? Despite this opposing view, also, they got the Nobel Prize together, right? Because uh, Santiago Ramon Cajal actually was able to, do his experiment, do his study, and, list whatever he listed, which at that time also most of the scientific community started believing. Right. And later, a lot of evidence was also given in support of it. Right. He was only able to do that because Golgi was able to create that state, right? So that is why collectively both of them helped in the advancement of this field. That is why they were given this Nobel Prize together, right? Now coming to Ross Harrison. Now this is an image from CSDN .net. This is Ross Harrison. He was basically an embryologist, and he provided support for the neuron doctrine in 1920s, right? Now, what he actually showed was that the dendrites and axons grow from the cell body, right? So earlier, Camillo Golgi had also observed that there are, longer growths that are axons, and reticular branch structure, which is dendrites that exist, right? But he said that they were a continuous network. Over here, he said that from a cell body, right? So, the neuron will obviously develop at some point, right? He was an embryologist, so he dealt with a lot of embryos. He was basically a developmental biologist, right? So he saw the development of a lot of these tissues, and he during that he actually demonstrated that these dendrites and axons are basically growing from the cell body, right? And they do so even when each neuron is isolated from others in the tissue culture, and it is not dependent on each other, right? If they were in a form of a network, then there would be one neuron, it will extend its processes, go and form another neuron, which will extend its processes, go and form a third neuron, so on and so forth. Right? So that was not the case. He proved that over here. Right? Also, he confirmed from Monica Hall's suggestion that the tip of the axon gives rise to an expansion growth cone, which leads the developing new axon to its target, either to other nerve cells or to the muscle, right? So this growth cone that exists in the axon is basically what is directing it towards other neurons or the muscles, right? Why is it going to the muscles? Why will a nerve cell or send its axon to a muscle? Because it will the nerve is eventually, or the neuron is eventually what is going to control the muscle, right? Control the movements in the muscle. So it needs to sit on the muscle to control the muscle, so some part it has to send over there, right? Even a puppeteer has to have those strings in his hand, right? Then only the puppeteer can control his puppets, right? Otherwise it will not be possible, right? So that is what uh, this example is, right? Then we can talk about Sanford Pillay, right? He also basically provided the final and the definite proof for the neuron in the favor of neuron doctrine. He basically performed well after the introduction of electron microscopy, right, in the mid 1950s. What he did was that he unambiguously, that is without any doubt, he demonstrated the existence of synapses, right? Specialized regions that permit chemical or electrical signaling between the neurons, right? So, in a reticular theory, there will be no synapses, right? Because everything is a connected network, right? So, once there is an origin point, that information will transmit through the entire network, right? According to reticular theory. But over here, uh, the existence of synapses means that there is gap between neurons, right? This gap is known as synapse, right? Again, when we study about the structure of the brain and structure of neuron, we are going to study about what exactly is synapses also. So just in case you are not aware of this concept, please don't feel left out. We will discuss upon it, right? Very soon we will discuss upon it. So, as I was saying, these synapses are regions between or gaps between neurons, right? And if these gaps exist between neurons, automatically reticular theory is proven wrong, right? Because it is not a continuous network, it has discontinuity between it, right? Then, again, this experiment also, maybe a lot of you have heard about if you have not directly studied about it. There was another physician and physicist, right, who was Luigi Galvani, right? He was Italian, and in the late 1700s, what he did was that he discovered the muscle and nerve cells produce electricity, right? So he was the first one to say basically that there is an electrical property in the muscle cells and the nerve cells that exists, right? This basically led to the beginning of physiological investigation of the nervous system. Right, so this was the experiment that he performed. What actually happened was, since he was a physician and he used to work with the a lot of model organisms, right? One of the common model organisms from the beginning has been the frog. Right? One day, what happened was that he and his assistant were actually skinning a frog, right? After dissection, they were skinning a frog, and what they found was that while they were doing that, although the frog had been dissected, the upper body of the frog was not even there. Only his only the leg area of the frog was available, right? Even in that leg area, there was some kind of a twitch, right? So if they accidentally touched the forceps over there, the leg twitched or there was a jerk in the leg, right? Now that kind of a jerk exists when somebody is alive, right? Over here the mouse is dissected, the posterior half is only present, the anterior half is not even there, right? So they thought that it might be because of some residual electric activity that is there, right? So what they did was they connected an electrode to the muscle of the frog, right? And you know that the hind limbs of the frog are very strong, right? So once they connected this electrode to the hind limbs, what happened was that like it will not always happen till like even 2 days after the frog was dissected, it won't happen, right? The electrical property will sustain for some definite amount of time, right? So after the frog was dissected, if the electrode was used to stimulate the leg, then what happened was that the leg twitched and it contracted and it folded like it would have happened in a living frog, right? Which basically suggested that the muscle cells and the nerve cells that are controlling the muscle cells have some kind of an electrical property, right? Yeah. So I hope that experiment is clear to you, right? Okay. Just give me one moment. Yes. Uh, so then we come to the discussion of these other scientists, right? They are Johannes Muller, Emil T. Boyce-Riemann, and Hermann von Helmholtz, right? Right. They were all German physiologists, right? So John Helmholtz and Raymond, all of them are basically German physiologists, and they succeeded in measuring the speed of conduction of electrical activity along the axon of the nerve cell. Right. Individually also they have done this experiment, and this is what they collectively, uh, you know, uh, kind of gave to science, right? That they succeeded in measuring at what uh, uh, speed there is electrical activity that is existent in the nerve cells, at what speed is it transmitted or is it conducted, right? Also they further showed that the electrical activity of one nerve cell affects the activity of an adjacent nerve cells in predictable ways, right? So for instance, I have 10 nerves that are arranged side by side like this, right? So they are arranged side by side like this. So if there is a stimulation in nerve number three, right, it is expected that the ones that are just next to it might also respond, right? So two and four might also have some residual response, right? It is not going to be the case that 2 and 4 did not respond at all, and when I stimulated 3, 1 and 10 responded, right? That is not going to be the case, right? So there is a very predictable way of stimulating activity of adjacent cells also, because it is an electrical property, right? Also, their work inspired modern electrophysiology in the 19th century, right? So, what is electrophysiology? As the name suggests, it has to do with, uh, you know, uh, understanding and sometimes even stimulating and recording the electrical activity of the body, right? It could be electrophysiology of the muscle, of the brain, of the heart, etc., right? So it entails basically both stimulating and recording the electrical activity of an organ, right? So let us study all of these one by one also. So Johannes Peter Muller basically gave the doctrine of specific nerve energies, right? Now it has been so well proven that it is known as a law of specific nerve energies, right? And he basically gave this doctrine in the 1835, right? So what he said was that the nature of perception is defined by the pathway over which the sensory information is carried, right? So what this means is that the mind has access not to the objects in the world, but only to our nerves, right? Which means that there are multiple sense organs in our body, right? If I am holding a tissue paper, right, over here, uh, okay, or a tissue roll over here, right? Uh, or something that is visible in this thing. If I'm holding this pouch again, right? So over here I'm holding this pouch, right? So because I can visually see that I'm holding it, right? My mind knows that I'm holding this pouch, and also because it was my mind's decision to hold this pouch, it knows that it is holding this pouch, right? And by touching it, I know the texture of this pouch, right? Uh, so my mind knows that it has a plasticky kind of feel it is not a cloth based material, etc., right? But it is dependent upon the path that my nerves are taking to record this. It is not like I am touching this, and some of the particles from this particular pouch are getting into my hand, so that my nerves are actually taking them in, and they are taking it to my brain so that my brain can see. No, that is not the case, right? So there is a certain kind of electrical activity that is being generated by the nerve endings in my finger when I'm touching that pouch, and it is going in my brain through a certain path. And my brain knows that once it is going through this path, this is what it means, right? It doesn't have access to this pouch. It only has access to the path through which the information has come to it, right? For instance, if okay, this is also actually a series of experiments that have actually been done, right? And in the 20th and 21st century only. So, for instance, there are a lot of odor receptors that are present. Odor receptors, matlab, something that will help you smell, right? There are so many of odor receptors that are present in the brain, right? So, if you basically smell something, right? It will fire certain neurons in your brain, right? And if we can record and we can have if we can record which neurons it is firing or which receptors that particular smell is going and binding to, and we can just stimulate those same receptors. Even if that smell is not there, your brain will, feel like you are smelling that same thing, right? So for instance, I like a particular perfume, right? And the name of the perfume is, say, Tulip, right? So I know that what that tulip, that perfume smells like, right? Uh, this is because whenever I'm smelling it, it is going, the odor particles are going and sitting on some specific odor receptors that are there in my nasal wall and my nasal epithelium, right? And that, receptor is basically transmitting that information to the neurons, which is taking it to the brain, and my brain is feeling that, okay, this is a good smell, right? Because it is coming through these neurons, it is a good smell, and I like it, right? Say that somebody recorded that, right? And was able to specifically activate the same set of odor receptors as was activated when I was spraying that perfume tulip, right? So even in the absence of that perfume tulip, if the same receptors are activated, I will feel like it is the same smell, right? Even though that perfume doesn't exist in this room anymore, right? So that is what the mind doesn't have access to the objects. It only has access to our nerves, right? What is the information is being taken to the brain, right? Then Emil Du Bois Raymond, right? He is renowned for co-discovering nerve action potential, right? So action potential is another very crucial part. We will study that very soon. It is basically the phenomena through which information is being transmitted from one neuron to the other neuron, right? Once a neuron is stimulated, it generates an action potential, and that stimulation is carried forward to the adjacent neurons, right? Through this action potential transfer. And he also pioneered experimental electrophysiology, right? So, who was Emil du Bois-Reymond? He was basically Muller's assistant in 1840, right? In the Department of Anatomy and Physiology at the University of Berlin. And they have co-discovered this action potential, nerve action potential, right? Again, Hermann von Helmholtz. In 1849, he measured the speed that the electricity flows in a frog's sciatic nerve. Right. So what he did to measure this was that he stimulated the nerve electrically at one end, and he measured the electrical signal at the other end, right? So that is what he did, right? And what did he find out? In case of this frog sciatic nerve, at least in the experiment that was done by Helmholtz, the speed that he measured was around 30 to 40 meter per second, I think, right? So once he actually recorded that, he thought that this is much, much slower than the speed of electricity, right? Although it is an electric signal only, but it is almost a million times slower than the actual electricity conduction, that signal, right? So why was that? Because in case of electricity, what is moving? Electrons are moving. Electrons move much faster, right? Over here, it is an electric signal, but what is moving? Electrons are not there, right? It is an electric signal that is generated by movement of ions, right? So since ions are moving, right, they are heavier. That is why this response is slower than the conduction of electricity, but it is still pretty fast. It is around 30 to 40 meter per second. Now we know that it can reach anywhere to around 100 meter per second also, right? Yeah. So that was the contribution of those three, right? Then there is another set of scientists, right? I think we can finish this today. So since I have spoken this long, I think I can finish it, right? Uh, okay. So pharmacology made its first impact on our understanding of the nervous system and the behavior at the end of the 19th century when Claude Bernard in France, this person, Paul Ehrlich in Germany, this person, and John Langley in England demonstrated that drugs do not just act anywhere on a cell, but rather bind discrete receptors typically located in the surface membrane of the cell, right? So through this, what was proven was actually that it led to the discovery that nerve cells can communicate with each other by chemical means, right? Because these are also cells, they also have receptors on their surface, right? So, based on which chemical they are releasing and which of the other neuron has the receptor for that chemical, they can select which neuron to talk to, right? So, they can communicate with each other by these chemical means. Then we talk about psychology, right? So, psychological thinking about behavior dates back to the beginning of the Western science when the ancient Greek philosophers speculated about causes of behavior and relation of the mind to the brain, right? So, one of the scientists was Rene Descartes. Right, so he in 17th century distinguished body and mind. Right, he said that body and mind are two separate entities. So this is basically known as a dualist view, right? In this dualistic view, he said that brain mediates perception, right? So brain is responsible for mediating perception, motor acts, memory, appetites, and passion, right? Everything that can be found in the lower animals. But the mind, which is the higher mental function, the conscious experience characteristic of human behavior is not represented in the brain or any other part of the body, but in the soul, a spiritual entity that communicates with machinery of the brain by means of pineal gland, a tiny structured middle of the brain. Right? So he said that brain and mind are different, right? How he described brain and how he described mind is also very similar to how we distinguish between these two, right? But he's when he said that both of them interact, he gave a physical point of interaction, right? Which is basically the pineal gland, right? This is a representation of the pineal gland, right? It is present in the midline of the brain. So if this is the entire brain, right? This is half of the brain that is seen over here. So this is the entire brain, right? It will be at the bottom in the center, right? Center bottom will be the pineal gland, right? So he thought because you know that the brain has two hemispheres, right? These structures are common in both the hemispheres, right? So he thought that this was the center point in the brain, and it did not have two parts. There was one pineal gland, right, which was located at the center, unlike other parts like cerebellum, thalamus, hypothalamus, pituitary, cortex, etc., hippocampus, etc. He thought pineal was only one and it was present at the center. So there was a reason that that was like that, right? And it was this gland which was basically connecting the mind to the brain, right? Because it was in the center also, it was a single part also, right? So this is the diagrammatic representation that he gave for this, right? This is the pineal gland. Sorry. Yeah. Now, however, this is the current knowledge that we have that yes, pineal gland is in the midline, bottom midline, right? But actually, if you zoom into this image, let me just show it to you. If you zoom into this image, you will see that there are two pineal glands, right? Just because it is in the center and it is small, he considered it as one. But it is also two, right? This is also bilateral, just like the brainstem, just like the cortex, just like the cerebellum, etc. Right? So pineal gland is also bilateral, right? Also, we know that this perception of his is untrue, right? That mind is talking to the brain through some specific physical entity, which is the pineal gland, right? That is not true. Then Baruch Spinoza, later in the 17th century, began to develop a unified view of the mind and body, right? So he had a dualist view, right? Now, Spinoza tried to develop a unified view, right? Which is what we are studying. We are going to study throughout this course, right? Unified theory, unified view of mind and body, right? That both of them are interconnected, interrelated, and interdependent upon one another. Then there were some scientists who were classified into these two groups, which were empiricists and idealists, right? So, who were empiricists? They believed that the brain is initially a blank state. They called it tabula rasa, right? That is later filled with sensory experience, right? Whereas what did the idealists say, right? One of the idealists is mentioned over here, Immanuel Kant. This is Immanuel Kant. He and other idealists believed that our perception of world is determined by inherent features of our mind or brain. That we are you know, uh, it is premeditated, it is predetermined as to how we are going to experience the world, right? That is what these idealists say, right? It is more likely a combination of the two. There is obviously some epigenetic thing that is also existent in forming our, rational, our reason, our experiences. But empiricist theory is more likely, or most of the part that is true, that with experiences with different, you know, uh, yeah, with different experiences, we initially uh, build up our perception of the world, our perception of what is what, right? So it is initially more like a blank slate that is later filled by sensory experiences, right? So we don't know when we are born what we will like to eat, what we will not like to eat, right? We might have eaten something sometime, right? We might not have liked it, so we don't like it anymore, right? Something of that sort, right? Also, it changes, right? With age, it might change, right? Maybe when I was 5 years old, I did not eat jackfruit, right? Now, at around 25 years old, I started eating jackfruit, right? So it changes also. So I think these are the final few slides for this history part, which I had given you a disclaimer already, was very theoretical, right? That is not how most of the course is going to be. But we have discussed the thought process behind it. We have discussed some experiments that were these, right? Or these statements. So I hope at least that was useful, right? And also you have understood why we are going through this part, right? So, in the mid 19th century, Charles Darwin set the stage for modern understanding of brain as the seat of all behavior. Right? How did he do that? He advanced the even more radical idea that animals could serve as models of human behavior. Right. So one did not really have to experiment on human or always keep looking at a human, because human has a larger lifespan also, right? A scientist or a physiologist or a physician cannot, keep looking at the person from when they are born to when they are 90, right? Just to understand what is happening to the brain with age, right? That person might not be alive that long, right? So he Darwin was a person who uh, rationalized that other animals that are similar to humans also can be studied for understanding of this behavior, brain, etc. Right? Thus, the study of evolution gave rise to ethology, right? What is ethology? Investigation of behavior of animals in their natural setting. And later, it also gave rise to experimental psychology, which is the study of human and animal behavior under controlled conditions, right? This is Darwin. You might all already know him, right? Sorry for the phone call. I am sorry. I can't find the call. Okay. Yeah. So at the beginning of the 20th century, this person, Sigmund Freud, introduced psychoanalysis. Right now, what is psychoanalysis? As the first systematic cognitive psychology, psychoanalysis is basically something that frame the enormous problems that confront us in understanding the human mind, like the problems that we discussed just prior to starting this history, part of the lecture, right? Then this person, Franz Joseph Gall, now he has done a lot of things and he was very adamant about what he was doing, right? Some of the parts that he said were true, some of them were utterly wrong, right? Just utterly incorrect also. So, in attempts to join biological and psychological concepts in the study of behavior, uh, Francis Joseph Gall, in early 1800, what he did was he proposed two new ideas, right? He was a Viennese physician and a neuroanatomist also, right? So he used to study the anatomy of the brain also, and he was a physician, right? He proposed these two new ideas. The first was that he advocated that the brain is an organ of the mind and that all mental functions emanate from the brain. Right? In doing so, he rejected the idea that mind and body are separate entities. Right? So he said both are like my brain is a sub entity of the mind, right? And anything that is being done is originating in the mind, it's in the brain itself, right? What was the second thing that he said? He also argued that the cerebral cortex did not function as a single organ, but the cerebral cortex contained within it many organs, and that particular region of the cerebral cortex controls specific functions, right? So it said that there was part of the cerebral cortex that was doing one region, one function, another part doing another function. It was not that whole cerebral cortex was involved in one or two functions only, right? So he enumerated at least 27 distinct regions or organs of cerebral cortex, right? Like this. Later, many more were added, each corresponding to specific mental faculty, right? So this is from Candle, this representation, and it shows an early map of functional localization in the brain, right? So he localized that this part of the brain is, uh, responsible for ideality, this is responsible for imitation, this is responsible for spirituality, for hope, for consciousness, etc. Right? Also, he assigned intellectual processes such as ability to evaluate causality, to calculate, to and sense or order, and to the front of the brain. Right? These abilities were towards the front of the brain. Then he said that instinctive characteristics such as romance, love, combativeness were assigned to the back of the brain, right? Now I will tell you the logic behind it that he used, right? Then he also said even the most abstract of human behaviors like generosity, secretiveness, religiosity were assigned a spot in the brain. Do you think that there is a part of our brain that is responsible for us being secretive, right? I would not have thought about that. But he went into that much detail and thought about that, and he even assigned a region of the brain for such a entity, right? Such an emotion. Although Gauss' theory of localization was prescient, right? His experimental approach to localization was extremely naive, right? So he has localized these brain areas, right? To different functions, right? But based on what has he localized them? That is a question, right? So what happened was he was in charge of a mental facility, right? In Vienna, I think. So over there, he used to study a lot of his subjects, right? And most of these subjects were people who were fixated on one kind of a thing, right? Somebody was very like people with OCD fixated on one kind of a thing, right? Or somebody who was very uh, scared all the time, or somebody who was very obsessively cleaning all the time, or somebody who was obsessively worrying all the time, something like that, right? So they were fixated on an emotion, and he used to study how their skull was, right? So what he thought was, I'm sorry, I thought I will sneeze. Yeah. What he thought was, or he derived a pattern, right? That people who are more cautious have this region expanded. People who are who are better at math have this region expanded, right? And this obsession of his basically began when he was in school, right? And and a lot of his classmates that used to get good marks all had, say, bigger foreheads, or they had, you know, bigger eyes. So he thought that this part of the head of there is bigger. So this part of the head is where their calculation skills, their memorizing skills, etc., they lie, right? So that is where it began. And these kinds of experiments he did, or observations he did on the people in that mental facility also, right, to map this kind of a structure, right? So he was influenced by physiognomy, a popular science based on the idea that facial features reveal character, right? So some people still do it, right? Because your eyes are like this. That is why you have a very loving nature, or because your eyes are not like this and instead they are like this, they are curved in this way, you look like a vicious human being, something like that. People still do that, right? Just for fun mostly, but he has actually been very serious about it. So, Gall believed that the bumps and ridges on the skull of people well endowed with specific faculties identified the centers for those faculties in the brain, right? Like I just talked about. He assumed that the size of an area of brain was related to mental faculty represented in that area. Again, we have spoken about it. Then his studies of localized brain functions led to phrenology, right? This science or pseudoscience actually was known as phrenology. That is pseudoscience because it was not true, right? Somebody would just look at this feature, oh, this area of your skull is expanded. It means that you are a con man, right? That is not true, right? So, a discipline concerned with determining personality and character based on the detailed shape of a skull, right? From the top, just by the shape of the skull, they are trying to decipher that, right? Later, in the 1820s, Gauss' ideas were subjected to experimental analysis by this French physiologist who is known as Pierre Florence, right? What Florence said was that he systematically destroyed Gauss' functional centers in the brain of experimental animals, right? So he took experimental animals, and for instance, he had the listed one region for alertness, right? He destroyed that region and he checked if the mice was more alert now or less alert. And it might not have changed, right? So what he said was Florence attempted to isolate the contribution of each cerebral organ to behavior, right? Whatever part he had said, he ablated one by one all those parts and he tried to check it. From these experiments, he concluded that the specific brain regions are not responsible for specific behavior, right? Or it would be possible that he had labeled the brain regions correctly, right? He was not accurate in that labeling. That is why the result that Florence got suggested that specific brain regions are not responsible for specific behaviors, but that all brain regions, especially the cerebral hemispheres of the brain, participate in every mental operation. Right now, this is what he said instead. We know that that is also not true. Any part of the cerebral hemisphere, Florence proposed, is able to perform all hemispheres function. Right now, this is what he proposed, but that is not true. Right. Then injury to any one area of the cerebral hemisphere should therefore affect all higher functions equally, right? But now we know there is a visual cortex area, there's a olfactory lobe, etc. Right? So if there is problem in that area, it will affect one kind of a function more than the other, right? So this was popularized as the holistic view of the mind, right? Mind as a whole is the holistic view. Then this holistic view was seriously challenged, however, in the mid 19th century by French neurologist Paul Pierre Broca, German neurologist Carl Wernicke, and British neurologist Hewlings Jackson. Right? Also, the regional studies of Broca, Wernicke, and Jackson were extended to cellular level by Charles Sherrington. Right now, this person is a great neuroscientist, right? We will study more about Charles Sherrington and about Wilter Penfield, about whom I have not mentioned in today's lectures. Both of these we will study in detail very soon, right? Okay. So, yeah, they championed the view of brain function called cellular connectionism. Right? According to this view, individual neurons are the signaling units of the brain and they are arranged in functional groups and connect to one another in a precise fashion. Okay. And then Wernicke's work and that of the French neurologist Jules Digerin in particular revealed that different behaviors are produced by different interconnected brain regions, right? So similar to what Jackson had done, there was a very famous Canadian neurologist, neuro surgeon actually, who used to work with a lot of epileptic patients, right? So in epileptic patients, what happens is sometimes the the epilepsy is arising in a particular region of the brain. At that time, science was not very advanced, right? So what they had to do in order for that person to live is remove that part of the brain. It was known as lobotomy, right? So I will just tell you so that I don't have to include it in the next class, maybe, right? So what he did was because he used to very regularly work with these epileptic patients, different ones of them, and epilepsy is something that could arise in different brain regions, right? So he had done lobotomy in different brain regions, and he had found after he would do that lobotomy, what he would see is that if this region of the brain is removed, this is the outcome that he's seeing in the patient. If this other region is removed, this is the outcome he's seeing in the patient. Also, what he did was that because it used to be an open brain surgery, you have to open the brain and you have to do the surgery, right? And you have to find that area also in that open brain which you need to remove, right? So you have to insert electrodes in the brain, right? So whenever he used to insert electrode, for instance, he has gone to an area which is responsible for sensing pain, right? So the person, although they don't have pain receptors over here, but is sensing pain in the fingertips, right? Or he has gone in, inserted an electrode in a region which is responsible for the person smelling flowers, right? So now the person is feeling like the person is smelling flowers just because that brain region has been stimulated, right? So these kinds of he used to insert it in different brain regions, also ask the patient what the patient is feeling like, note it down, make notes. Based on that, also a lot of understanding of the human brain has come forward. Okay. Yeah. So that is it for today. And I hope that your fellow batchmates who were not able to join, probably because of this miscommunication regarding the link, I have recorded the class for them, right? I hope they'll join us on Sunday. I hope so will you. And having said that, thank you a lot for having attended the session. I would really appreciate it if you guys were more vocal, right? Even if you want me to, teach something specific in the next lesson. Or talk about detail about something, right? I also might not know a lot of things. I might also need time to prepare, so I'm willing to do that if you would like to, understand something in particular or something like that. So, anyway, thank you for now for attending the lecture. I hope you learned something, and I hope that you are much better at understanding of neurobiology once you are done or through with this lecture series, right? So thank you for now. I'll I'll stop the recording. Thank you.